And I'll just confirm with you that you can see my slides. Oops, one moment.
Yes.
Yes, we can see it.
Perfect. One moment here. All right. All right, I'll just start going. So again, my name is Jordan Pomeroy. I'm a medical officer and clinical team lead in the Division of Cardiology and Nephrology at the U.S. Food and Drug Administration. I do have a scientific background in pluripotent cell biology, as well as have completed a postdoc in cardiac tissue engineering. So I do have some context for all of the wonderful development that's been discussed here. Again, this format for the in silico part, it seems like it's an emerging field, and I can't wait to see how this works in terms of our regulatory framework. But exciting to see all the work that's being done, and I congratulate you all on that. Okay, so again, as a FDA, I just want to make sure that it all is clear here that anything I say should not be construed as formal regulatory advice, and that if you do seek formal advice, that you should come to us through formal channels. But always happy to hope that what I talk about today is helpful to your development programs, and I have no disclosures to report. So I was tasked with giving kind of a clinician or a clinical reviewer perspective for this in silico integration of new approach methodologies. And so I'll do my best to do that, but I really want to focus on specifically in my role as a medical officer, considerations of CV toxicology or CV safety, which are paramount to our early review of drugs as they come in as investigational new drugs, either through the PIND stage or during active development, the IND, and then finally, of course, for new drug applications. But if you think about the overall spectrum of CV safety, is that you actually have many different mechanisms of potential CV toxicity, and we found these across multiple different drug development programs. But as I heard earlier in the talks yesterday, is we're really thinking about animal models and how to replace animal models through some of our efforts using new approach methodologies. Um, it's hard to capture everything, I think, within a single NAM. And I think we heard about scale of the cost associated with a single animal versus the overall utility of single chips or microphysiological systems. We're not quite there yet, so I think it's hard to consider using this for recapitulating everything that we do with our preclinical animal models. But of course, targeted context of use become important as we think about all these different areas that drugs could have CV toxicities. And we know about this because of also all the different classes of drugs which have hit these different mechanisms. So we're talking about really complex biology as well as pharmacology as we move forward in these drug development programs from CV toxicities perspective. Um, Quickly, just thinking about the overall spectrum of drug development from preclinical to clinical development, early on, the bulk of the work that's done is through our preclinical animal testing to understand potential adverse effects in the cardiovascular system. And as we progress in our clinical data capture, we use that information to help inform how we're going to monitor for cardiovascular safety within the context of these programs. And if we see a cardiovascular signal in the preclinical development, oftentimes that doesn't mean we can't move forward. It means we need to understand how to deploy specific monitoring for that identified signal. And that's really important for early development, but also we can have CV signals that come up that we're not seeing in preclinical development that are coming on during clinical development. And it's important to think about how NAMs or our preclinical development can be used to de-risk any of these clinical signals that are seen. And I really want to highlight that in specifically for CV toxicity or cardiovascular safety, that NAMs really do have a role across the whole spectrum. And I really think if we can't think about replacing all of animal testing, we can think about this as a place where we can do focused de-risking of signals that we've seen, and that provides an opportunity to define a context of use. Uh, Before I go on, I always like to go back to the regulations and the guidances. We have our regulations which allow us to ask developers to look at specific cardiovascular safety questions. And those regulations really come from 21 CFR 314.50, which talks about the content of a drug application. And then as for biologic, it's 21 CFR 601.2. Now, while those are kind of just a bunch of numbers, what they do provide is the actual legal framework for the FDA to ask for specific batteries of testing. And this comes in with our guidances that were developed in accordance and interpretation of these regulations. And those become important through S7A, which discuss the ability to request safety for cross different organ systems, including CV safety, and looking at specific areas of CV safety, in this case, repolarization or QT or pro-arrhythmic signals through S7B. And then M3R2, which is really important for those slides I showed earlier with the different spectrum of CV toxicities that have been observed, is that this provides for the battery of preclinical testing that is required. Now, as we think about moving forward in time, the question is how will these guidances be refined to include new approach methodologies? And so again, the challenges and questions that we face from a regulatory perspective is, can these NAMs replace the full spectrum, and will the regulations or guidance change to reflect that over time? So we work in a kind of static system, but of course at the FDA, we're working to try to figure out how to deploy new tools to be able to answer these different questions of CV toxicity. And I'll highlight some of those right now. I think Ksenia Blanova did a great job talking about iStand and the growth of iStand over time in her talk yesterday. But again, recently in the last year, it's almost a year old now, we have the roadmap to reducing animal testing. It's following the 3Rs principle for reduce, refine, and replacing animal testing. And a lot of that has gone to cell-based systems, but of course, we're seeing here in just the talk preceding is that there's this potential for a large breakthrough for the use of in silico modeling or artificial intelligence enhanced tools to understand preclinical risk stratification. But the FDA, the long-term goal, which has been publicly stated, is that in the next 3 to 5 years, we would like to largely replace our I guess historical precedent of using animal testing to guide our clinical monitoring strategies, and whether we can replace that in large part by new approach methodologies. One place where we've already launched this is within the scope of monoclonal antibodies. December 2025 guidance basically talks about how we can remove animal testing from preclinical testing of monoclonal antibodies. And part of this is because we have a lot of understanding that the specificity and the inherent biology of monoclonal antibodies is different than some of our small molecule compounds. I do point out to this reference here as well, which was recently published by many in our Division of Cardiology and Nephrology preclinical testing group, but it talks about the actual experience with new approach methodologies at the FDA. And one of those areas that it's used in silico modeling is the comprehensive in vitro pro-arrhythmia assay, or CIPA. And in this, it really looks at cell-derived as well as in silico modeling to predict potential QT prolongation or pro-arrhythmic risk. And so you can see that there are some limited cases where we've deployed this within our regulatory framework, but it does show that there's ability to get these in early in our processes so that we can improve them over time. And then for NAM qualification, I think Ksenia's talk yesterday on iStand really laid out, how you get into this process specifically for microphysiologic systems or complex in vitro models. I think iStand is the place to go, and you can see the growth of that through the applications over the past year. And also, I think in silico NAMs may fall within the scope of that. It's a good question. I don't know if Cassini has had any in silico NAMs. I think she said there was a few that came in the pie chart that I saw yesterday. But obviously, there's the drug development tool program and the MDDT over on the device side as well. And then these collaborative workshops and forums such as the CPATH group, as well as the NIH Tissue Chip Forum here, are a great place to seek some of that early consensus on how to move these new technologies forward. So the question that I always get asked is, qualification required for any of your NAMs to come to me within the Division of Cardiology and Nephrology for proposal? And the answer to that is no. We will, if you have an idea and you've defined a context of use that is supporting a specific drug development program, then you should come talk to us about it. It's always better to do that early, but you do not need to get qualification through iStand or DDT to be able to introduce that concept to us. I will say, if you think that your NAM or tool is going to be useful across multiple different drug development platforms, the qualification does provide an avenue to decrease the need for that engagement across those different programs if we have an understanding of its context of use, and that's been validated and gone through the validation programs at the FDA. One area where we see this is human-induced pluripotent stem cell-based cardiomyocyte assays. Within CEDAR, this is about a year and a half old, this paper now, but there's been over 100 different applications that have included NAMs that are based on human-induced pluripotent stem cells, either as single-cell models or complex models. But none of these were really validated coming in, and so we do see these as part of our preclinical packages as we review drug applications. So in conclusion, as we think about the overall complexity of cardiovascular toxicity, um, even the non-clinical animal models don't fully cover all the places that we would expect to see CV toxicity based on our knowledge of prior drug development programs. So there's a large space where we can use NAMs to understand specific areas that we observe either in later clinical development or in this case maybe predicted toxicities so that we can perform de-risking early on to help our understanding for the initial clinical studies. FDA is putting a lot of effort behind stating that we want to see new approach methodologies presented to us for review so that we can both understand it from a regulatory perspective to educate us on the use of these, but also how to implement them and improve the efficiency of implementation. We've seen most success so far through microphysiologic systems or complex in vitro models that have used human stem cell based platforms for cardiovascular toxicity testing. But it's very clear that in silico models are going to be a large place where are either complementary or supplant some of these models themselves. So I do thank everyone for their time, their engagement, the very exciting development programs that you have, and I look forward to seeing them here at the FDA.